So today's lesson is going to introduce concept 4.3 in Unit 4. It's about the cell cycle and introducing mitosis. So let's ask ourselves the question, what makes cell division so important? Well, for unicellular organisms, they're going to depend on cell division to make an entirely new organism. So that's a lot like the asexual reproduction that we saw. But for multicellular organisms, we depend on cells to divide for basically three things. We can summarize it. Cells need to divide for reproduction purposes, and cells need to divide for growth and repair. So for reproduction purposes, we're going to talk about a special process called meiosis. For growth and repair, and to actually become a multicellular organism, we're going to depend on a process called mitosis. Now, these both exist with a cell cycle, and you can kind of think of a cell cycle as a life cycle, kind of like we're born and you grow up and then you die, and it's a cycle, and that's what keeps happening. And this is virtually a circle. But to do all of these things, no matter if it's reproduce, repair, and grow, the cell has to make a copy of its DNA, make extra organelles, and gather extra materials, sort of like what you would need to do before you go on a trip. So we're going to take a look at this picture, and I do have all the writing here from the class. We're going to just focus here. The place where it kind of starts is G1 phase, stands for gap 1. And gap 1 is part of interphase. So you'll see that here in purple. Gap 1 phase is when the cell's growing. I added the words doing its job. It's doing its job. Now, sometime the cell will go into a phase called G0. And G0 is a permanent state of doing its job. The cell really isn't getting much bigger. It's doing its job. Sometimes the cell stays in G0 or it comes back into this cycle. We can't really tell the difference by looking at these cells in a microscope. When a cell has made the commitment to divide, it's getting too large and it's not as efficient. The next phase will be the S phase, or the synthesis phase. This is where DNA replication happens. The next step is G2 phase, preparing to actually divide. So gather new organelles, because if it's going to divide into two, it's going to need separate sets of organelles and materials. So now that's interphase, G1, S, and G2. And you really cannot distinguish the difference between these other than if you would look at the DNA amount. So for example, if this is humans, there'd be 46 chromosomes here, 46 here, but when DNA doubles, there'd be 92, because it's 92 or is 46 times 2. So DNA, that's what is happening here. The DNA is replicating. So now you have 92. You're going to have 92. So the only way, again, you'd be able to distinguish between G1 and S is the number of chromosomes. Then we're going to have 92 here as we go into the M phase. And this is cell division. Mitosis consists of prophase, metaphase, anaphase, and telophase. And then cytokinesis is the other part of cell division. And then when you end up, you're going to have 46, and 46 are going to be two identical cells. So taking a look at the G1 portion, and I have those chromosome numbers here. We did take some notes about what happens in G1, what happens in S phase. You may want to pause the video and copy the notes. We also took a look at chromosomes. So if you have not read 4.4 in the textbook, you want to read about the chromosomes. Lots of vocabulary. Chromatids, chromosomes, centrioles, centromere. It can get very confusing. What we have here is a chromosome that has replicated, and we can tell it's replicated because it's attached together at the centromere. And that centromere then has these spindle fibers, or small microtubules, that are also attaching to the centromere. And the centrioles found in animal cells are going to produce these spindle fibers, and they're going to be responsible for kind of setting up and organizing where these chromosomes go. Now, within these chromosomes, you're going to find a segment that these are genes. So there's a gene here, there's a gene here, a gene here. So the genes continue all along the chromosome, and the copied one would have exactly identical copies. So after that little brief sidestep to look at chromosomes, we came back to look at G2 phase, where a cell is going to also be preparing to divide. And I showed students what real life pictures of an animal cell and a plant cell look like in interphase. And again, it's just doing its job, so it spends 90% of its time here. You can see a nuclear membrane. You can see chromatin. Since this is an animal cell, you can see the cell membrane. In a plant cell, you can see a distinct cell wall. Well, when a cell gets ready to divide, things start to look different. So if you compare this picture and you go back and you look at this picture, you can tell the differences. One of the things that happens is the nuclear membrane disappears or dissolves, and the chromosomes start to appear. Metaphase, the sister chromatids are lining up in the middle, and spindle fibers start to appear. In anaphase, the sister chromatids start to pull apart. And in telophase, which is kind of like the end, the chromosomes are regathering and the nuclear membrane starting to appear. So that was all nuclear division. Prophase, metaphase, anaphase, and telophase, that's nuclear division. But the cytoplasm has to split as well. So that's what we call cytokinesis. The plant has Golgi vesicles that are carrying cellulose to build a new cell wall, whereas an animal cell is going to pinch off. It creates a cleavage furrow to create two new cells. So the next step is to actually look at a slide of an onion root tip. And we pick an onion root tip because a root is quickly going to have a lot of cells and cell division. So we can actually look at an area that's rapidly dividing, and we can try to capture, what are some of those cells? What phases are those cells in? And we're also going to practice, can you identify what phase they're in? Those are the common things. You should also be able to make sure you could list them in the correct order from beginning to end. And that's where we're going to stop.